Everyone, in this video we will be discussing about one of the sequencing method, that is Maxam Gilbert sequencing. This sequencing method is also known as chemical degradation method because the nucleotides which we want to sequence, they are degraded by certain chemicals in certain bases. In particular bases they are being degraded, and the bases which are degraded, they are chemically modified. So let us see what are the reagents required for this reaction to take place. So the main thing which we require during any sequencing is a template strand. Whatever template strand is given, we have to denature it because we need it in a single strand. Before that, if someone is giving us for sequencing one sample, so they are in the double stranded form. So this double stranded form first of all must be denatured into a single stranded form by supplying heat. Or the other thing which we have to do is, first of all, we have to do a PCR reaction, PCR, to amplify this double stranded to get multiple clones. Then we should denature it into a single standard form to perform our reaction. The other thing which are required during this reaction is alkaline phosphatase. What is the work of this alkaline phosphatase? It removes a phosphate group in the 5' end. In the 5' end, there is a phosphate group present, as we have discussed earlier. So this alkaline phosphate has to remove that phosphate. Next comes the phosphorus 32. It is a radio label. Along with the ATP, it is being attached in the 5' end. The other chemicals which are required in this reaction are dimethyl sulfate. This dimethyl sulfate add a methyl group on the guanine. The next is formic acid. The work of formic acid is that it breaks the glycosidic bond. Next is hydrogen. The work of hydrogen is that it works only on pyrimidine. That means it splits the ring of pyrimidine. The next is NaCl. The work of NaCl is it covers the thiamine base, and whenever it is working with hydrogen, only the hydrogen can work on cytosine. We will be very clear when we see the experiments how it is taking place. So here we should know that the work of hydrogen is that it works on pyrimidine. That means it splits the ring of pyrimidine, and the work of NaCl is it covers the thiamine base pair. So hydrogen will work only on cytosine. Next is a piperidine. The work of piperidine is that it breaks a sugar backbone whenever there is a modification of some of the nucleotide. Now step by step we will see how this process is taking place. We have already known what are the components or what are the requirement for this reaction. The next thing is that how this process is done. This is the first step. So as we have discussed that we need a DNA. Suppose we are having one of the DNA, and this DNA is converted into a single stranded form. What we are having a 5' end and a 3' end. In the 3' end there is a OH group present, and in the 5' end there is a phosphate group present. So the first thing is that we have to remove this by the help of one of the enzyme, that is alkaline phosphate. What this does is it removes the phosphate group from the 5' end. The next thing what we have to do is we have to add a labeled 32 phosphate. This is radio label. So why do we need a radio label? To identify the 5' end, we are doing this radio labeling. So if this radio label is been added, like for the removal we need one of the enzyme, that is alkaline phosphatase. For giving or for adding also we require a enzyme. What is the name of the enzyme? It is the polynucleotide kinase. Along with radio labeled ATP, this polynucleotide kinase will help in adding the phosphate, radio labeled phosphate, in the 5' group. So what is the next thing? We are done with this. The next thing is that is the addition of the chemicals. So as we have discussed that we require some of the components for this process to be done. So after adding the components into each tube, each appendage of tube, we will divide the chemicals into 4 tubes. That is for 4 nucleotides to be sequenced. In the first tube, what we will add is we will add one of the chemicals that is known as dimethyl sulfate. What is the work of this dimethyl sulfate? This dimethyl sulfate will change the guanine into a methyl group. That means a methyl group will be added in the guanine that will be modifying the guanine. In the next tube, what we will add is we will add formic acid. Formic acid. So what is the work of this formic acid? The work of formic acid is that it will break the glycosidic bond of only specific nitrogenous bases. So here it will work only for adenine as well as guanine. In the next tube, what we will add? We will add hydrazine. Hydrazine without NaCl. Okay, so what is the work of this hydrazine? The work of hydrazine is that it will work only on pyrimidine bases, and it will break the ring of the pyrimidine. In the next tube, what we will add? We will add something for cytosine. Here we will add hydrazine plus 1.5 molar NaCl. So what is the work of this NaCl? We have already discussed that the work of hydrazine is that it will work only on the pyrimidine base and it will split the ring into different parts. So here what we are using? We are using hydrazine as well as 1.5 molar NaCl. What this NaCl does, it covers the thymine base. So now it cannot work on thymine. It will only work on cytosine. Here it was working on cytosine as well as thymine. But whenever we are adding the NaCl, it will work on cytosine. So let us see how it is done. Here we can see that we have given the chemicals. We have given the chemicals. Along with it, we have given the piperidine. Piperidine plus chemicals. So we are getting the degraded DNA. The next step is that we will perform a gel electrophoresis. So here we can know how it is being interpreted. So you can see here that it is always analyzed from the downward position. So here you can see that there are 4 wells and the chemically degraded solutions are loaded into the gel side by side. So here you can see that the bases are present here and here. So it is for guanine. This is for guanine. Okay, we can see that the first one is here. It is for G and this is for A and G. Okay, in this case, as these two are common for AG and here G is common, so it is for G. The next we can see here that it is for C plus T. But here we don't have any band. If it would be C, here we should get one band, but here we don't have any C. So it is for T. Here also it is for T. The next here it is A plus G. In case here what we got is we got G because the gel was showing one picture on G. But here it is A plus G. That means it is knowing inside, it is knowing not knowing for any G. So it is A. Here we can see that along with A plus G, G is also coming. So it is G. Here C plus T as well as C is coming. So it is for C. The next is C plus T. Here for C it is not coming. That is the reason it is T. Here we can see it is A plus G. Here G is not coming. That means it is A. Here again we can see that C plus T as well as C. That means this goes for C. Okay. This is how we can interpret this. So the sequence for here is G, T, T, A, G, C, T, A, C. As we have discussed in the Sanger sequencing, we were getting the complementary strand. Here we are not getting the complementary strand in this Maxam Gilbert sequencing. We are getting the original strand. It is in the 5' to 3' direction. Okay, let us again interpret this data with other diagram. We can see here that it is having one of the fragments. This fragment is G, C, T, A, C, G, T, A. It is from 5' and in the 5' and it is radiolabeled. And in the 3' hand it is present A. So when we cleave for A plus G, we will get certain bands. You can see here that if it is cleaved here, we will get GCT. If it is cleaved in some other region, that is here near G, if it is cleaved, so we get GCTAC. Until this we get the sequence. If it is cleaved near this region, there will be no cleavage, or it will only the A will be cleaved out. So we are getting the sequence until T, that is GCTACGT. The same for G. Whenever we are getting a cleavage, suppose we are getting a cleavage upon this G is modified and we are getting a cleavage over here. So that so in that case we will not get any of the bond. Where is the next G? Here we are having the next G. If the bond will come and cleave here, we will get a sequence, that is G, C, T, A, C, until C we will get. The next is for cytosine. If we will get a cleavage over here in this region, then we will get only G. The next C is here. So from here we can get G, C, T, A. This is the sequence which we got. Next is C plus T. Here we can get the cleavage for C as well as T. So here let us erase this. So in case of C plus T, here we will get one cleavage so that we get only guanine. If we will get a cleavage over here, we will get guanine and cytosine. If we get a cleavage over here, so we will get guanine, cytosine, thymine, adenine. If the cleavage is here, along with cytosine, we will get one guanine. These are the sequences which we got. These are small fragments which we got. And in gel electrophoresis, what we will do is we will add this, we will load this side by side. Okay, so this is in the 5' to 3' direction. So it is arranged in 5' to 3' direction. So here we are getting here a band for cytosine as well as cytosine and thymine. The common one will be written here. C is common here. So here we can get cytosine. Next here is cytosine plus thymine. That means no cytosine is present. So it will definitely code for thymine. The next is adenine for guanine. In common, what we have guanine. So here guanine is not present. It is showing only adenine. So here it is A. The next is cytosine and cytosine plus thymine. In common, what we have cytosine. So it is coding for cytosine. Here we can see it is coding for A plus G as well as G. So in common, we have guanine. That is the reason here guanine is showing. Next is C plus T. Here we don't have anything for C. That means it is T. Here we are seeing that it is A plus G. It is not showing anything for G. So we are having here adenine. This is the way how the sequencing is done. So let us again see how this process is being done in a detailed form. So the first thing which we required is we require some of the components, that is template DNA, alkaline phosphatase, phosphorus 32 for radio labeling. The chemicals which are required are dimethyl sulfate, formic acid, hydrogen, NaCl, piperidine. The first step is that we have to remove the phosphate group with the help of the enzyme alkaline phosphatase. The second thing is that we have to add one of the radio label tag in the 5' end. The third step is that we have to give the solution or treat the solution with certain chemicals for G, A plus G, C plus T as well as C. After that, we have to add the piperidine for the cleavage. After the cleavage has been done, it will be radio label, the 5' radio label fragments will be separated out, and we will run it on gel electrophoresis in different ways for getting the data interpretation. So this is all about the Maxam Gilbert sequencing, and if you like this video, please do like, share and subscribe. Thank you.